Jayakar, and thank you very much, Doctor, for giving us a clinician's perspective. But I'm going to introduce to you right now Dr. Greg Frederick. He's a professor here from, he's in the Department of Biology and Kinesiology. I owe a lot to him because if it weren't for him, I wouldn't be here. But I think one of the interesting things about Dr. Frederick in what he's about ready to provide for us is his intimate knowledge of viruses. When he came here, he introduced a very novel way of teaching freshman biology, and that's using bacterial viruses potentially as therapeutic agents. We're dealing with an antibiotic-resistant world right now. And where do you get those viruses? Well, you got to go find bacteria that have viruses on it. And Dr. Frederick, with collaboration with Dr. Ballerine, introduced this to our Gen Biological Sciences 1 and 2. So I know that our biologists that get educated here know a lot about viruses, probably more than they want. But to be fair, this is one reason why I think we're really privileged to have a true in-house expert. He has been researching, because I've been talking with Dr. Frederick in the hall for the last nearly 2 years about tracking this COVID-19 thing, and I'm always learning something. So I would like you to put your hands together and thank you, Dr. Frederick, for gracing our stage.
Thank you, Dr. Dyer. It's good to be here with you guys tonight, and I look forward to talking to you. I know there are people in the audience, digitally, out virtually in the world, and around that maybe don't know a lot about LeTourneau University. So I just want to tell you, I've been here for 7 years. This is my 8th year here. And LeTourneau is an incredible university. We do give our students excellent opportunities to learn things that they wouldn't otherwise get to learn around the world in other universities. And our students have been very much involved in the last 5, 6 years in going out into the environment and isolating environmental samples and bringing those samples back into the laboratory, and then isolating viruses from those environmental samples and looking at those viruses to characterize them. We teach them how to grow the viruses. We teach them how to extract the genomes from the viruses. We teach them how to analyze the genome so that they can compare the genomes of the viruses that they've isolated and characterize those at the genomic level. And a lot of the skills that they're learning in our freshman biology classes are very much related to the skills that are being used to understand this current coronavirus crisis and what's going on with it. In fact, I have a group of students that are working pretty much full-time in my laboratory on the coronaviruses, and a number of other students that I'll introduce to you a little bit later have been looking specifically at the spike proteins for these viruses that we're all concerned about. The title of this session and Thursday session is Seeking Clarity and Spreading Hope. That's what I want to do tonight for you all. I think just those word clouds that we did at the beginning where people said frustration and confusion and tired, those were words that I think we've all felt in the middle of this pandemic. And there were other words that I didn't get to jot down on my pad, but they were there, and you felt them too, and I have as well. And so hopefully this session will give you some information that you can use to make some decisions that will really guide you in the process. I've told you our students get incredible opportunities here at LeTourneau as scientists, and I think we grow some of the best scientists in the nation that go on to medical professions and other areas to serve humanity. But they also learn something I think even more important than that. They learn to develop a relationship with Christ, and they realize that Christ is the most important thing to them. There are just a couple of, did I blank us? Oh, we're back on Dr. Oh, I figured it out. The remote goes the opposite of what I think it should. Thanks. So clarity, I thought about that, and what are the synonyms for clarity? These are some that I came up with. The first one was transparency. If we think about clarity, it means that we can see clearly, and we have to have transparency. The next I thought about was tangibility. We have to be able to grasp. We have to be able to understand. We have to be able to comprehend if we're going to have clarity, and clarity should give us all of those things. So when I was thinking about transparency, I was thinking about this verse in Psalms 139. It says, "Search me, God, know my heart, test me, and know my concerns." We've all had concerns in the middle of this COVID pandemic. "See if there are any idolatrous ways in me, and lead me in the everlasting way." God's beseeching us to seek him so that we can understand the world around us. As we think about tangibility, this verse in Colossians came. It says, "Be careful that no one takes you captive through philosophy or empty deceit, based on human tradition or based on elemental forces of the world, and not based on Christ. For the entire fullness of God's nature," that's the world that we're living in, dwells bodily in Christ. And in comprehensibility, I ask God to do this for me. Open my eyes so that I may contemplate wonderful things from your instruction from Psalms. And as I thought about hope, optimism was one of the first synonyms that I came up with. And now may God, the God of hope, fill you with the joy and the peace as you believe in him, so that you may overflow with hope and by the power of the Holy Spirit. That's what we all need. And I think that's what Scripture tells us we need, is that we need to have that joy. And we need to have that hope. And I hope these sessions will help us to have that. And I thought about, too, when we think about hope, we have to have some kind of promise that we can hope in. And so this promise that we have from God is that we can have eternal life with him. And I just want to make it clear to you, I am a scientist, and I'll explain some of my background to you, but if we don't have that understanding of eternal life, if we don't fully understand the hope that he's given us, then all of the rest of the things you're going to learn tonight are going to be meaningless, because without that hope of eternal life that we get through Jesus Christ, our life really is meaningless. So I hope you see that in the process of this presentation. And then faith. This is kind of some instruction for us. This is actually Jesus speaking, and this is Jesus right about before he was to be crucified. He said, "Your heart must not be troubled." Other translations make that a commandment. Do not let your heart be troubled. And if God gives us a commandment, Jesus said, "If you love me, obey my commandments." So I think we want to obey his commandments. And he says, "Your heart must not be troubled. You believe in God and you believe in me. And I am the way, the truth, and the life, and no one comes to the Father except through me." If you believe that, then your heart shouldn't be troubled no matter what your life circumstances. And I just want to start tonight with these thoughts about hope and with clarity. Now I want to tell you a little bit about myself. My students know me pretty well because they get to spend maybe more time with me than they wish they did. But my first job was for this company. And many of you don't know Sanofi, but Sanofi, pre-COVID, was actually the world's largest vaccine producer. And when I took my first scientific job with them after my bachelor's degree, they asked me to work in one of their vaccine production facilities. And the facility was involved in producing vaccines, and they put me in the virus section for quality control. Since COVID, they're maybe not quite the largest vaccine producer, but after that, I learned a lot about vaccine production. I learned a lot about quality control. I learned a lot about ways that we can make mistakes in vaccine production. My next job was actually doing pharmaceutical research with Merck and Company, and it's currently one of the largest U.S. vaccine producers, and I'll talk to you, their name will come up again a little bit later in the process. I did those areas of research, and I learned a lot about pharmaceutical chemistry, a lot about virology, a lot about vaccine production, and then I went and did a Ph.D. at the Kansas University School of Medicine in the Department of Microbiology, Immunology, and Molecular Genetics. From there, after my Ph.D., I was recruited to a pharmaceutical company. You all may not know Novartis, but again, pre-COVID, it was the second largest pharmaceutical company in the world. It's in Basel, Switzerland, and you may have heard of Hoffmann-La Roche, or Roche now. And they were the largest pharmaceutical company. They're right next door to Novartis in Basel, Switzerland. And then from there, I was recruited to join the faculty at UT Southwestern Medical Center in the Department of Pathology. And there I worked on a number of molecular diseases and trained medical doctors and residents and others in the areas of pathology. From there, God kind of changed my trajectory, and he sent me to the world's maybe second most populated nation in Asia to teach at a medical school there and serve as a missionary there as well. And in the meantime, he's done a lot of things in between. But that's just a bit about me, and I don't want to spend a lot of time there, but I've got some knowledge in these areas. So I've got to ask you a question, and I don't have it built into their Slido, but my question is, do you trust science? And I think that's a good question. I think that's a changing question for many Americans. Science is a process, and I trust processes as long as processes are designed well. If an auto manufacturer is going to make a car and they do everything right, I'm going to trust and I'm going to drive their car. But if they don't do the right quality control, and I have recalls every year and maybe even have an accident because of some of their lack of quality control, then I may not trust their car the next time. But science is just a process. And science is done by people. Scientists are people, just like you. You probably realize you've made a mistake along the way at some point in time. I have too. Scientists are people, and they make mistakes as well. Some of you may know that the first diagnostic that was sent out was actually produced by the US CDC for the COVID virus. And they made a PCR test kit so that we could detect very minute amounts of the virus in samples that we acquired from patients. There was just one problem with those early test kits. In the process of producing those test kits, they actually contaminated the reagents of the test kit with the genome of the virus. So in the first few months of the tests that were coming back, many of the tests were coming back positive because the genome was actually in the test kit. And unfortunately, people, as we teach our students, ran negative controls and realized, hey, wait a minute, we're getting positive results without putting any specimens into this test kit. So we know scientists make mistakes. Even the CDC makes mistakes. They demonstrated it for us in this particular crisis. Both science, if the science is not conducted well, and if the experiments haven't been based on good observations, they make mistakes, and scientists themselves can make mistakes. So I'm just curious again, how closely have you been following the literature for COVID? I can't do the Slido, so if you'll just raise your hands, those are here. How many of you have been watching the COVID literature and reading the COVID literature? Can you raise your hands? I see some hands that are being raised. That's awesome. Hopefully you're not reading mainstream media, but you're reading the science that's published. I did this scan just a few days ago on the 15th of this month. There are already 4,430,000 publications with the word COVID-19 in them. Those are published publications. That's actually more publications. If everyone in Texas had read one of those, there would still be about 400,000 of those that didn't get read. That's a lot of publications. If we just look for the virus name, there's over 1 million that have the virus in the publications. I haven't read all those. I suspect most of you haven't read all of those either. It's really hard to follow the science because the science is so abundant. And in reality, most of the science that was done 5 months ago isn't published yet. And you aren't able to see it unless you look at preprints and read those. But it's a lot to read, and it's certainly a lot to trust if you're going to look at that much literature. It's a big problem. We've got an elephant in the room. And it's this disease. It's this virus. And it isn't a small problem. As was mentioned, I imagine many of you have family members who have experienced COVID. And probably a lot of you have family members who have died from COVID. And certainly you have family members who have been sick. And so it's a huge elephant that's in the room with us right now. I want to pause for just a second because I do want to give you hope in the process of this message that I'm going to deliver to you. But I need to say this. I was talking to a friend, and I consider her a dear friend, and she's a healthcare professional. She's a clinician, and she had been fully vaccinated. And I found out that she was experiencing COVID. And so I contacted her, and her symptoms were very severe. And I told her that I'd really like to talk to her more and hear about it. And And in the process, she asked me why she could have COVID when she was fully vaccinated. So I began to explain to her some of the things that I'm going to talk to you about tonight and why she could be so sick with COVID. And in the process of me explaining the science behind the vaccines and why the vaccines aren't working as effectively as many times you hear in the mainstream media, in the end of the conversation, she told me, "You've caused me to lose my joy." That was certainly not my intent. I wanted to help her understand the science, and that's the same job that I've been asked to do tonight. So I don't want to help you lose your joy. I just want you to understand the science, and I certainly don't want to offend you. So if you've lost family members in the process of this pandemic, then I feel your grief too. In the last couple of weeks, actually last couple of months now, I lost a couple of very close friends that had other comorbidities, but they did die with the virus in their system. And so we're all feeling the grief from this, and it's a huge elephant. The disease is that COVID-19, and this is the virus that causes it. And we're going to talk about the SARS-CoV-2 virus, and we are going to get to the vaccine. These are numbers that I pulled for Texas. There have been over 4 million reported cases. That's why I could tell you that there's about 400,000 of those articles that wouldn't have been read if each one of these individuals that's been infected or at least diagnosed as being infected in Texas. And that's the number of deaths total. And I think these are, again, of about 3 days ago that I pulled these numbers. And so in Texas, we see about a 1.6% death rate from the virus. But the literature is pretty clear that even in the elderly populations in assisted care facilities, about 30 to 50% of the individuals who are infected with the virus are actually asymptomatic. So if we take the high end of that, we've got a 0.8% mortality rate from the virus itself. It's probably in reality somewhere in between. If we look at the last 7-day average, we see some numbers that are significantly different, but that's because of something that's happening with the variants that are out there right now. If we look just at Gregg County, that 526, I can remember it because that was the street address and the house that I grew up on. That's the number that was up there a couple days ago. I think it's a few higher than that. That's the number of deaths in Gregg County since the beginning of the pandemic. And in Gregg County, it looks like we've got something more significantly higher going on than we have in the rest of the state. So one thing I want to tell you is there's something that's bigger. I think fear is a bigger elephant than the virus itself. The verse I showed you before told us that we're not supposed to fear, but so does Isaiah 41. Isaiah 41 has another commandment. I've pointed this out to my students a number of times because when I was in seminary, I had to go to seminary. I left my position at Southwestern Medical Center to go to seminary before I could go to that big country in East Asia. I decided I wanted to know all of Christ's commandments, so I started making a list. This one isn't one of Christ, but it's a commandment that's in the Bible. It says, do not fear. God tells us we shouldn't fear because he's with us. We shouldn't be afraid, God says, for he is our God. If he's your God, you shouldn't be afraid. He says he'll strengthen you and he'll help you and he'll hold you with his right hand, his righteous right hand. If we believe that, then we shouldn't fear. Here's why we shouldn't fear. I've told my students before, on a good day, on a healthy day, when you're healthy and you have no disease, your body needs to produce at least 60 million immune cells for you to stay healthy. That's the number of immune cells that need to be produced on any given day when there's not a disease state for you to maintain your health. And we know very well that fear and stress lower immune function. If you've ever had an outbreak of shingles, and many of you in the audience are older, you may have, but even in my young students, when they get to finals week, many of them experience an outbreak of shingles because they get stressed, and fear causes stress, and stress lowers the function of the immune system. And so if you want to stay healthy, listen to God's commandments. Don't fear, because stress and fear lower the function of your immune system. Dr. Jayakar did a great job of introducing some of the pandemics that we've had before. This is one of the most successful vaccine campaigns that we've had, and you can see a lot of death from smallpox. And this data is very explicit in what's going on here. By the end of the 1800s, 1900s, we were seeing very little smallpox. But prior to the introduction of the vaccine that Edward Jenner, Dr. Jayakar mentioned, we were seeing about 1 in 13 people who were infected by the smallpox virus actually dying from the virus itself. And Edward Jenner, an incredible scientist, saw a group of people who weren't being infected by the smallpox virus, and he said, what's different about their lifestyle? And he realized that they were becoming exposed to what we now know is the virus that cows get that can cross-react. This is one of the reasons we needed that campaign. This is a young boy who's infected with the smallpox. You can see that it's very deforming if it doesn't kill you, and it causes a lot of problem. This is the virus itself. It's a double-stranded genome virus, and one of the things that's key about this is that for the smallpox virus, there is no animal reservoir. We don't know of any other animals that the virus can jump out of humans into and then live there for a while and come back into humans. So it's been successful for a number of reasons. One of the reasons, as my students know, that even though genomes of viruses that are double-stranded can mutate, they mutate much more slowly than other types of viral genomes. This is about the polio vaccine, and for those of you that are my age or older, you probably have some friends or remembrance of friends who had experienced polio when you were growing up. And we introduced a very effective vaccine for polio back in the 1960s, and maybe just a little bit earlier than that. We have 2 forms of vaccine for polio now, and they work very well, but they're not without problems. And this graph, I don't know if you can see it well, but this plot right here, it shows the reduction of cases of paralysis due to the natural wild polio vaccine. This is just about 2 decades that you can see here, the most recent 2 decades. This is about 27 right here. And this is the number of cases of paralysis that have been induced because of the vaccine itself. And one of the things that we're unfortunately seeing with the polio vaccine is that there's a sharp incline in the amount of paralysis that's we're seeing because of the vaccine, even though the amount of paralysis of the wild-type virus has become very low. Again, this virus doesn't have any known animal reservoirs. The next one, Dr. Jayakar mentioned, this one is the measles as well, and I'm kind of jumping through some of those visual slides so we can get to some data here to talk about. And so this is the measles campaign, and you can see the measles virus there. Again, it's got a negative strand, single-stranded RNA genome, and again, no animal reservoirs. One of the things that most who've studied a general biology course know is that when DNA is replicated, the mechanisms that replicate DNA have very strong proofreading and very strong error correction. So if there is a mistake that's introduced into the copying the genome, it's very quickly fixed, and very few of those actually pass through to the progeny. But the enzymes that replicate RNA don't have the same kind of proofreading, and so we tend to see a lot more mutagenesis introduced into those viruses just through the natural replication process. So we've got some successful campaigns in DNA viruses and RNA viruses, and you know that the SARS-CoV-2 virus is a single-stranded RNA virus. This was just for fun because Dr. Seuss got a little bit of grief in the last few months, and so I put this in here, but it actually looks a lot like a virus. We've got, especially those envelope viruses that we're looking at tonight, we've got the membrane that's here, we've got some funny looking proteins that stick outside of it, and most of them have some other proteins down inside, and usually there's some kind of message or genome that's down inside of there. And we can all think thoughts, and hopefully we can learn from it. Hopefully that's what we're doing right now. This is the guy we're talking about now, the SARS-CoV-2 virus. It's a coronavirus, and most of you have had coronaviruses throughout your life. You probably had 10, 15, 20, or 30 coronavirus infections in the process of growing up. This guy's just a little bit different. He seems to cause more disease than those viruses that you've experienced before, and we do have some different breakdowns of the virus categorization, but this is the general structure, and this is the This is the spike protein that you see right here. All of these big proteins are the spike protein. The spike protein interacts with the ACE2 receptor on your cells, and that's one of the ways of entry into our cells. What we can also see is the genome's actually inside, and it's wrapped in protein, and it's called the nucleocapsid protein. On the outside of the virus, besides the spike protein, there are two other prominent viruses. One is called an envelope virus, and one is called the membrane virus. We use M as a designation for the membrane and E for an envelope. And those are two prominent viruses that are on the surface of all coronavirus, two prominent proteins on the surface of all coronaviruses. The problem with the coronaviruses is there are many animal reservoirs. So even if we vaccinate the human population, coronaviruses all can jump back into animal reservoirs. In fact, there are many studies that show your favorite cat or dog, or if you have a large cougar or a lion at home, they can all be infected by the SARS-2 virus, and we see that happening over and over again. This is maybe a better representation of the SARS-CoV-2 virus, and you can see, again, the spike proteins sticking out here. The predominance, big proteins here are all the spike proteins. But one thing, the reason I included this picture is I wanted to show you that the spike proteins are not uniformly coating the surface of the virus. In fact, there are many other proteins. The membrane proteins are down here, and the envelope proteins are down there. They're depicted slightly differently, but I'm not going to show you that. But when you're infected with the virus itself, you actually get antibodies to the spike protein. You get antibodies produced to the M protein. You get antibodies produced to the envelope protein. You get antibodies produced to the nucleocapsid protein. The other thing is when you're infected by the virus is actually mutating inside of your body. So you're not just producing antibodies to one spike protein, but you're producing antibodies to many different variants of the spike protein as it replicates down inside of your body. I've had students, 12 this semester, doing research with me. We learned a lot about bioinformatics in my lab and looking at structures. This is actually the spike protein here. It's shown in more molecular form that you can see. We'll see if this is going to play. I want you to see it. So I guess it's not going to play tonight for me, but this is a structure, and if we rotated it 90 degrees with the right-hand side toward us, you would actually be able to see the receptor binding domain. And that's the part of the spike protein that binds to the receptors on our cells that allows it to be able to infect in our cells. And I wish it would play, but it's not going to. So we'll go to this slide. We've got this pandemic, and we think we're fighting a virus, but what I want to tell you tonight is we're not fighting one virus. We're actually, at this moment, at least 3-4 days ago, fighting about 3.9 million different viruses. My students in my class isolate viruses from the environment. We sequence the genomes, and you're hearing about a few variants, but there are actually 3.9 million different SARS-CoV-2 viruses that have been isolated. We group them into different clades so that we, with our finite minds, can understand these viruses, but there are that many of them that are out there in the environment that have been isolated and sequenced and characterized. It's a bigger problem than you think it is. And so this is a picture. If you remember, we introduced a vaccine right here between March and April, and it's been introduced at different rates, as Dr. Jayakar mentioned, across the world. But what we're seeing is early on, pre-vaccine, we had a lot of the alpha variant, and it's very closely related to the original Wuhan isolate. And then we had some other minor variants that are up here, and we didn't see much of the Delta variant that we're hearing about in the news right now. There's a little bit, less than 5% there pre-vaccine. As we move toward the vaccine, what we see is these graphs are a little bit hard to read, but for example, if we're looking at the alpha, you can subtract from this number down here, which in my eyes looks like about 18, up to this number here, which is about 35, and it gives you the percent of that particular variant that was in the population. And as we introduced the vaccine right about here, we saw a very quick shift in the types of variants that were being isolated in patients toward the Delta variant. This blue area is all Delta variant, and now we're seeing almost 100% Delta variant in patients that are being diagnosed with COVID. But it's not just the Delta variant. We forced the Delta variant to mutate, and now this part of the plot right here is all what we group in the Delta variant, but there are many different forms of the Delta variant, again, because the antibodies are selecting for different variants in our population. And the vaccines are actually pushing those variants that aren't as well bound, don't have as high affinity to the antibodies to be able to survive and to be able to pass on. There's some other characteristics of virulence that play a role in that, but it's obvious that the vaccine being introduced right at this point is when we start to see shifts in the number of different variants that are out there in the world. Dr. Jaikar did a good job about talking about the different types of vaccines. Actually, Johnson & Johnson, I mentioned Merck earlier, they have some vaccine facilities. In fact, one of the vaccines is the Janssen company in the Netherlands, and they're the ones that develop their vaccine, but they don't have production facilities. So they're actually working with Merck to produce their vaccine. And the other vaccines are very similar, but we do have some experience. Moderna's been around for 11 years, and they've been trying to do things with messenger RNA modalities for quite a while. They've tried to use it for cancer therapy. They've tried to use it for other therapies, and so far they haven't been able to get it through FDA approval because of a number of complications. And it wasn't until this particular application that they were able to push through. They changed their name once, and so if you go look at stocks, it's only been around for 3 years. But if you do a little bit of research, the name change, it'll take you back to the origins of that company. It's a US-based company. Pfizer's international. Pfizer's vaccine's produced in Germany, and you might have just read that the UK is not accepting the Moderna vaccine anymore. They say they have plenty of the Pfizer vaccine, and you kind of have to wonder, is it because it's produced in Germany, or do they think it's more effective? We'll talk about that down the road. Johnson & Johnson is actually produced around the world, but the subsidiary that developed it is actually a Dutch company. All right, so only the Pfizer vaccine's been FDA approved, and you guys said you didn't really care if it was FDA approved, so I'm gonna skip through these slides. At least that's what I got from the previous survey that was done, but most of them just have this emergency use authorization. The media is saying they're approved, but they're approved for emergency use authorization, and emergency use authorization basically says if there's no other treatment that's available, that's effective, that's been approved. And so that's a big question mark at the end of all of that. So now we go to the question of whether the vaccines are safe, and we have to look at different aspects. And I was a little bit perplexed by your response to the FDA question, because I think FDA approval is important. FDA approval is what keeps companies from producing products that could cause injury to you, that could cause cancer in you, that could cause other disease in you. And so from my personal perspective, I think that FDA approval is very important. When I started my Ph.D., I was in a virology lab working on a virus that causes cancer in humans, and my Ph.D. advisor went to work for the FDA, and unfortunately he couldn't take any students with him. But personally, I think that we need some organization that protects us from things that might be released as products to keep us healthy. So back to the vaccines, are they safe? And what I want to do is look at a little bit of data. I'm not going to take you to the CDC website because you can all go there, you can read the things that you're going to see there, but I'm going to show you some primary data from around the world. And this is just a study that was done. It was a quick survey that was done. This survey was actually done in June of this year, so it's about 4 or 5 years old, or 4 or 5 months old. But it was done by the Association of American Physicians and Surgeons. It's, I guess, the largest community of doctors in the U.S., and they saw that 80% of the people who hadn't been vaccinated, and at that time it was nearly 60% of all physicians and surgeons, they said that they believed that the risk of the vaccine outweighed the disease. There's obviously been some persuasion for them since June of this year. A more recent study done by Northwestern University shows that 27% of healthcare professionals are not fully vaccinated at this point in time. So there's been some shift. We just saw this in the media, and I thought I would throw this in here because if the mandates stay in place, we're going to have some serious problems with travel in the U.S. 40% of the The TSA in the U.S. hasn't been vaccinated yet, and the government's saying that they need to be. They should have already started the process. That's going to cause some very serious problems. So this is the real question I think you want to know, and we're going to consider this here. And again, I'm going to go back to this. Whatever you do, don't be anxious. In everything, by prayer and supplication, and with thanksgiving, let your requests be known to God. What I'm going to tell you to do in the case of COVID is what you should do every day of your life. Communicate with God, be educated, pray to Him, meditate on the question that you have, and I believe through Scripture He will provide an answer to you. So I'm not going to answer your question. I'm going to let you decide, but I'm going to give you some information that you might be able to decide with. My story is, in May of this year, I did have COVID. I'm 63 going on 64. My COVID, praise God, was very mild. I probably, if COVID hadn't been in the media, would not have known that I was infected by the virus. I did lose a little bit of sense of smell, but it was pretty much a bad allergy day other than losing a sense of smell. I probably would have been at work, and so maybe it's a good thing I knew. But my wife and I had a vacation scheduled to the Caribbean and actually Mexico. And so I was wanting to go on that vacation. So I was very optimistic I was going to get well. And I spent a lot of time in the sunshine because sunshine is very important for the absorption of vitamin D. And you need vitamin D to replicate those 60 million cells every day. And so you need to eat well. Dr. Jayakar said that as well. And I tested again in less than 10 days, and I was already PCR negative for the virus, and I got to go on my vacation. That's my story. So what should you do? The first thing I'm going to suggest that you do is find out if you've already been infected, because I said even in facilities that are extended care facilities for the elderly, the surveillance studies that we've been doing are excellent because we can see very clearly that 30 to 50% of the people that are infected don't have any symptoms. So if you don't know that you've had the disease yet, you could very well be sitting here and have antibodies to the virus. And a recent study that was just submitted for publication from Israel shows that those that have been infected by the virus itself have 12 to 15% times stronger immunity than those that have been vaccinated. So how do you do that? How do you know if you've been infected by the virus? If we were having this session a month ago, I would have told you quickly go to this site and sign up for this study that UT Health Science Center in Houston is doing, because they're doing a pretty large study of about 85,000 people around Texas looking to see who has and who hasn't been infected by the virus. And you could have signed up for free, and they would have tested your antibodies against COVID 3 times. But unfortunately, I looked last week, and they've already closed their cohort. They're not accepting any more participants at this time. But it was a perfect study, and I told my students, did you guys sign up for it? I hope you did, because I asked you to, but some of the others in the office did, so if you didn't, I still love you. Yeah, so it's a great study because it's a longitudinal study. They ask you a whole bunch of questions. Do you know that you've been infected by COVID? And if you do, when was it? And have you been vaccinated? And if not, why not sometimes? And then they start testing your antibodies every 3 months. And it's a quantitative PCR test, so you can see exactly how many antibodies they have. The goals of this study is actually to estimate how many people in Texas have been infected by the SARS-CoV-2 virus. They want to estimate how many people have been vaccinated, and the antibody test can actually tell that. And they want to know how many of those develop antibodies. That's critical. I'll tell you why in just a little bit. They want to know how long the COVID-19 antibodies last in everyone's system once they have been vaccinated or infected. And I want to identify the characteristics of people who have developed antibodies. And they also want to know about the people who haven't. One of my good friends, I think he might be listening right now, I'm not sure, he works for a major airline. He comes in contact with thousands of people every day and tens of thousands of people in the course of a month, and in a very confined space. His fiance also had a case of COVID, and so I suggested that he go sign up for this study, and surprisingly, with all the exposure that he's had, he doesn't have any antibodies to COVID yet. There's a couple of reasons why he might not. We don't always develop antibodies to infection. The first line of defense when there's a viral infection is that a group of cells in our body called natural killer cells recognize those cells that are infected in the early days of infection. They play the role in the first 7 days or so. If they see the cells that are infected by the virus in the first 7 days, their job is to go kill those cells. And if they're effective at killing those cells, you won't produce antibodies. That's part of the innate immune response. And we see people that have a strong enough innate immune response that they don't go on to develop disease and they don't go on to develop antibodies, because the first steps of the immune system that we don't hear anything about in the media is actually doing the job that God designed it to do. It's dealing with the virus infection before the load of the virus gets too large and out of hand, so that we have to develop the adaptive immune response. Developing antibodies, if you've never seen the antigen before, initially takes about 7 days, and the response is very, very low compared to later response times. And so we need that innate immune system to deal with those types of infections. It's happening all the time. If you've taken part in this study, here's my results right here. It's a quantitative test, and they'll tell you, yes, you're positive for antibodies to the SARS-CoV-2 virus, and they actually tell you how many units of antibodies that you have. So when you go back 3 months later, you can actually compare your amount of antibodies in the serum to what you had seen in the previous months. And they're doing it over a total of 6 months. I didn't get into this cohort until 5 months after infection. And so this first test was 5 months after I'd cleared the virus, and the next one will be 8, and the next one will be about a year afterward. But most of the studies in the literature are showing those that are naturally infected, even if they're asymptomatic, have antibodies that endure at least that long. So that was for the spike protein, but these are antibodies to the nucleocapsid. Remember when I showed that picture of the virion, and you could see down inside the genome, the nucleocapsid protein is the protein that protects the nucleus. And they also look at the nucleocapsid protein, and you can see also I have a pretty good titer against those proteins. That's the analysis that tells me and tells them that I have been infected by the virus. Because if you've only been vaccinated, you only have the first class of antibodies. You won't have this class of antibodies. So if you're in that test, and I know a few people who are, in fact, that friend that I just mentioned a bit ago, he doesn't show any antibodies now. His company's mandated that he be vaccinated, so he chose a vaccine to use, and I'm excited to see what his titers are when he goes back in. I'll find it very interesting if he hasn't actually produced any antibodies to the vaccine itself. Some people don't. So knowing that cohort had closed, I actually tried to call the lab, and you can try it too. It's Clinical Pathology Labs on 4th Street, just north of Walmart. I wanted to ask them how much it would cost if I walked in to get that antibody test, because it's a very informative one. It's actually made by Roche, that company that's in Basel, Switzerland, that I mentioned previously, that was right across the way from the company where I did my postdoc. But it tests both of those antibodies to the spike protein, antibodies to the nucleocapsid, so you can see whether you've been vaccinated or not. You probably want to go get that. Here are some other places that do it. These guys are going to want money, and I didn't have time to call around, but the typical test is somewhere between $50 and $100, and it might give you some hope. It might give you some peace of mind if you're not sure you want to take the vaccine, to know whether you actually have antibodies or not. Again, because many studies are coming out that are showing antibodies produced by the viral infection are actually more protective than the vaccine itself. So back to this question. I saw a meme that just looked exactly like this, and I couldn't believe it was real. So I actually went to the CDC website, and I pulled all the data that they have on the CDC website. The meme, I think, went back to 30 years. I decided 2 decades was enough data to look at, so I started back in 2001, a little preview, and started in 2001, and I just told the system to tell me about all the deaths that were associated with any vaccine that had ever been administered in the US from 2001 to 2021. And we have had deaths, that's not in question, but the question is, this number is pretty disproportional to all the other years. Nearly 11,000 deaths have been reported. Now, opponents to using this kind of data will say that those deaths haven't been confirmed, but if you take that as a given, then the lack of deaths there also hasn't been confirmed. But the number of reports is obviously different from the previous years. And this is a CDC website data. If you look at the number that are hospitalized, we see a very similar picture. The number, again, jumps for this year, and that goes up to about 35,000 that have been hospitalized after the vaccine. This is not after the virus, this is after taking the vaccine. You can go do those types of searches. My brother doesn't have a degree in science, and so I asked him, hey, can you go do this? And sure enough, he could do the analysis himself. So if you want to confirm the analysis, then go and look at that. Here's one of the concerns. We are getting a lot of people that are vaccinated, and that may not be a bad thing, but even as early as September of this year, we're seeing studies that say that vaccination doesn't correlate to the amount of disease that we're seeing. And this study looked at 68 different countries, and in the US, they looked at 2,947 different counties. I don't have time to go into all the data, but the reference is there. And by the way, I got one request previously for references. If you would like those, I'm gonna give you my email address at the end so you can send me an email so that you can check out these references as well. And this one's an interesting one. This one's just this month, and it's in the New England Journal of Medicine, and basically we're already seeing that the vaccine antibodies are not protecting individuals. This one's in Qatar, but there's many countries that are showing similar types of data. The last one is a very interesting. The UK was very proactive in getting their vaccines delivered to their community, and they actually do a surveillance study, and they post the results of their national surveillance study every week. And so I just want to walk you through a little bit of this data. We're saying that we need herd immunity. That line was yellow when I looked at it on my computer screen, but that line shows you where we need to have herd immunity based on the current definition. That's 70% of the individuals that have been vaccinated. This is data again for the UK, and it's by age, and I don't think you can read those, but they started their vaccine campaign with the elderly, so this is 80 to 84, and we get younger. They don't allow 16-year-olds to be vaccinated and younger, so they're down here on this line, and I think this line is 16 to 18, if I remember right. And they started the vaccine campaign and allowance for vaccinations based on those that are most predisposed to COVID, so they started with the oldest age groups. But what you can see here is they very quickly moved toward herd immunity after vaccination. Okay, this is just vaccine uptake, but they showed basically they reached 95% vaccination in most of those age groups pretty quickly after they mandated that people be vaccinated. If we go to blood donors in the UK and look at those that have antibodies, we see that, I can't even read those numbers, but now I think I can. Right here, we've got 16 weeks after the start of the vaccine. They've reached herd immunity, and now we're out here at about week 40, and we can see that herd immunity was reached, and so things should be good in the UK, right? They've got herd immunity, they've got over 70% easily, and now it looks like they're close to 90% with immunity to the SARS-CoV-2 virus in the UK based on this. Oh, and this assay, by the way, is showing you what's going on with the spike protein and what's going on with the nucleocapsid protein, so we can see which individuals have been infected by the virus and which individuals have likely received the vaccine. If we look at different age groups, again, this is sero prevalence in blood donors, so we're looking at those that donate blood. We again see that we very early on reached that herd immunity state. This is about 12 weeks after the initiation of the vaccine campaign, that in the UK they reached what our current definition is of herd immunity. For the younger groups, it took a little bit longer, but the reason it took longer was the younger groups weren't allowed access to the vaccine. But again, if we look at the data, by week 24, we had herd immunity reached in the younger groups of individuals in the UK. So that's very encouraging. It is saying that the vaccine is inducing antibodies. And we knew that. We saw that in the FDA trials, and in my mind, I wish that Pfizer had allowed their trial to go on, or that the government had allowed it to go on for the full additional year of trial, because I do think that that is important. So if we take those same numbers from that same study in the UK and we look at which individuals are actually being are actually having COVID infections based on unvaccinated to vaccinated, we see something very interesting. And this is divided by age groups. This is a graph I had to make, but this is the rate of infection per 100,000 based on the unvaccinated status, and this is the infected numbers. And you can see at the young age group, there are more unvaccinated that actually contracted COVID, but we see something very curious in the other age groups. As you go up the age groups, you actually start to see more vaccinated people contracting COVID than unvaccinated people in this national study that's done by the UK. By the time we get to the ages 40 to 49, it's over twice as many vaccinated people are contracting COVID as not. And in all the age groups above that youngest age group, we see more vaccinated people being infected by COVID than the unvaccinated. That's a very perplexing data set. That has to make you wonder about what's going on. Now, in all honesty, they did see a slightly higher hospitalization rate in the unvaccinated data sets, and they also saw a slightly higher a higher death rate in the unvaccinated. They saw something else that they didn't analyze, and I wish they had. They didn't look at those rates in those that had been infected by the virus and those that had received the vaccine to do a comparison that way. That would have been very telling. But still, in all the age groups except for the youngest age group, there was a higher infection rate in the vaccinated group than the unvaccinated group. And so that makes us wonder, are the unvaccinated really the dangerous spreaders? Because the vaccinated individuals tend to be less symptomatic and they're being infected more frequently according to these numbers. You have to ask that question when you look at data that's like that. So there's a lot of studies, and I just threw this back out there. The one we just looked at is this one. We discussed these guys previously, but I wanted to use this slide to refocus for a little bit. Again, that data that I just showed you is data that's published by the UK government and their surveillance studies. This is another study that came out. Some of the therapies that we've tried to do is produce monoclonal antibodies to some of the variants that are coming out. And as early as July of this year, we're already seeing that many of the monoclonals, this is talking about one specific one, doesn't block the Delta variant anymore. And the Delta variant, at least in some cases, is evading the antibodies that are produced by natural infection and by vaccination. That's getting pretty concerning, and I think you saw why in one of the previous slides. So I tried to think about why we might see the data that we just saw in the UK study, and I wanted to walk you back through. This is the basic strategy for mRNA vaccines, and Dr. Jayakar mentioned this previously. We put a mRNA into a lipid nanoparticle, our lysosome. We introduce that, and hopefully it's going to be picked up by some cell. In this case, it's an antigen-producing cell, and this is the normal process that induces immunity. The mRNA is used for translation, and a protein is made. In the process, this antigen-presenting cell is signaled because of the presence of that mRNA. I don't have time to teach an entire immunology class, but there are a lot of proteins that are made by cells that are called toll-like receptors, and toll-like receptor 7 binds to single-stranded RNA. And when it's bound by single-stranded RNA, it signals that there's a problem. It signals that, hey, we need to do something about that, and some cytokines are produced that signals the production of additional protein on the surface that then interacts with the antigen-presenting cell to antigen-specific T cell, and that's the initiation process of making antibodies. That's the normal route. So in the mRNA vaccines, we have the signal, we have the mRNA that's introduced, and we make the protein that we want the antibodies to be produced against. But now we're talking about, in fact, since the beginning of the year, we're talking about using the same strategy to fight against autoimmunity. And this looks pretty similar. If you look close at it, there's only one difference. In this case, the mRNA that signals, hey, there's a problem isn't present. And in this case, we can induce tolerance to a given protein. And so if you have an autoimmune disease, your body's making antibodies to some structure in your body, and that's not a good thing, causes many different diseases. And so the companies that made these vaccines are trying to figure out a way to use the strategy. In fact, that's been their strategy all along, was to use this therapy to try to fight autoimmune disease. The only problem is when we introduce the mRNA disappears from the system very quickly. It's very labile, it's broken down quickly, and what remains is the protein, the spike protein that we're producing. And so we end up with cells that are antigen-producing cells that are introducing the spike protein. And I suspect in a few months we're going to begin to see a lot of tolerance that's being introduced in the human population. What tolerance does is says, this is a protein that you as the immune system should not respond to, and that's going to be a serious crisis if we do see that. We're already seeing them developing strategies to use this to fight autoimmune disease, in other words, to induce tolerance in other diseases. And I won't be surprised because of the delivery mechanism and the lack of the messenger RNA after a few days that's stimulating that danger signal for the cells to go into this pathway and enter into this pathway. We're going to have to see. I don't like to be a prophet, but that's my prediction at this point, and I think it begins to explain some of the information that you just saw in the UK study. All right, that was pretty depressing. I might have just stole your joy, and I've only got a few minutes left if I have that many, so thanks for being patient with me. And I do want you to find joy. Am I pushing the wrong button again? We'll see. Let's look back at this guy. Let's trust God. And we can put faith in vaccines, but there are some antivirals that we use nucleoside analogs that are using with some promise. We've got monoclonals, but we're already starting to see, I put still in small letters there because they're becoming less and less effective as the viruses mutate. There's a lot of studies around the world that actually showing that ivermectin is effective in treating COVID. And I was excited to see that just this month, the Texas Health Science Center got a multi-million-dollar grant to actually study the efficacy of ivermectin in the treatment of COVID. That's an exciting thing. There are currently over 20 countries that are using ivermectin to control their COVID crisis, and hopefully the U.S. will realize that there's some validity to that. So again, go get checked for the antibodies. You may already have the antibodies, and if you do, you're likely very good. The CDC will tell you that you will develop stronger immunity to the spike protein if you do take the vaccine, and from an immunological perspective, that's true. The problem with the vaccines are they only produce antibodies that circulate in the bloodstream. They don't produce the antibodies that are on the mucosal tissue. And since this virus enters the body through the mucosal tissue, those viruses that are in the bloodstream don't protect you from infection until the virus is already circulating in your body. Most of all, I want you to get peace from Christ, because that's where our true peace comes from. And this verse in John basically says, your peace I leave with you, my peace I give to you, not as the world gives. The world wants to give us peace through certain mechanisms and processes, but Christ's peace is very different. Our hearts should not be, they must not be troubled, and they must not be fearful. Those are words from Christ, and that's what I hope for you, is that you'll somehow figure out a way to deal with the fear that this pandemic may have caused in your heart. Because again, Christ knew what he was talking about. We know now as scientists that fear and stress greatly diminish the function of the immune system, and we all need a healthy, functional immune system right now. As I said, I've got students that have been working with me too that have been working hard all semester, and then these guys. If you see these a name that you know, go ahead and applaud and cheer for these guys. They've been working hard to help me with some of the 3D modeling of the spike proteins and looking at the different variants and trying to determine which parts of the spike proteins might lead to different variant and being able to escape the antibodies that our bodies are producing in response to the COVID vaccine. So thank you very much. I promised you that I'd give you my email address, and there it is. If you've got questions, you can ask me your COVID questions. That's a real email address. I created it. Google hadn't allowed someone else to choose it yet. So email me any questions that you might have, and I don't know that I'll check it daily, but I'll try to check it at least once a week with questions that you might have, and maybe we have a few questions or time for those digital questions that you all might have.
Yes, thank you, Dr. Frederick. As before, we have lots and lots of questions.
If I don't get to them, just email me at that email address.
Certainly. So we'll ask just a couple here because we're almost out of time. If mRNA codes for parts of a spike protein, let me put this up, sorry. See if that works. There we go. How can we really keep up with virus variants?
That's a great one. So it depends how we bend the rules of the FDA. We could today produce another mRNA that codes for the Delta variant, and we could put it in lipid nanoparticles pretty quickly, but we don't know, and typically we won't allow that to happen unless it goes through FDA clinical trials. FDA clinical trials, and even in the rush setting that we used for this particular case, took over a year to get through all of the trials. And the trial set was small, and you already know that they cut the trial off a year prior to when it would normally be. So we would have to develop a lot of different ones, or the other option is to do what viruses do and actually make lipid nanoparticles with multiple variant mRNAs encapsulated in those lipid nanoparticles. Again, you have to ask yourself, is that a good idea or is it not? It's increasing the variables in the experiment, and we don't know what kind of results it might have.
Thank you. And we have time for one more question here, and this one has to do with treatments. As you can see, I'll let you read that one.
Other treatments like monoclonal.
Is that about me?
Oh, you can read it too, Scott, yeah.
I think I got it. So
Okay,
they're basically
good.
asking about other therapies. One of the things that we see with no matter what type of respiratory infection it is that many times the virus doesn't cause the severe complications. It's actually the secondary infections by bacteria. So the azithromycin, it's misspelled there, but azithromycin is an antibiotic that controls a lot of respiratory infections. And I honestly think that's a good idea. It's not a good idea to give everyone, but if a person's having severe COVID symptoms within the first 3 or 4 days, I think they should be put on azithromycin as a prophylaxis so they don't have a secondary infection, because the bacteria that populate the lungs can't be removed because of the damaged tissue that the virus is producing. There are other types. Ivermectin, I don't know, the studies are mixed on that one. And so I'm going to leave that one for some other authorities. You can go do some research. But when I looked into ivermectin as a possible therapy, it's really quite mixed. And I hope we do a larger scale clinical trial. But I know that people are urgent about trying to figure out what's going on with this. Monoclonal antibodies are great. We can make monoclonal antibodies pretty quickly. If you've been infected by the virus, they can actually go and harvest your plasma cells that are producing antibodies. They can fuse those with a myeloma cell, and they can immortalize those plasma cells from your body, and they can make very specific monoclonal antibodies that produce specific antibodies. But those monoclonals have to be analyzed too. They have to be tested, or they should be, to make sure that they don't bind to things that they shouldn't indiscriminately and cause other problems. What we do know is when antibodies bind to things, other cells come and tend to kill those things that are bound by antibodies. So we want to be very careful that the antibodies that we're using don't actually bind to other components of our cells that might lead those cells that are destructive to actually attack our own tissues.
All right, let's thank Dr. Frederick again here.
Could I get you to just put my email address back up just in case they didn't get that? I really would love to answer any questions that you all have. I'd be glad to do that. If it's possible, but if it's not, it's just yourcovidquestions@gmail.com.
Okay, Fro, could you put on the picture for the panel discussions? All right, this was part 1. Part 2 is this next Thursday. It's going to be a very, very different event. This is going to be a panel discussion where we're going to have Drs. Kimberly Quiet representing nursing. We've got her husband, Jeffrey Quiet, representing counseling. We got John Barrett, who's going to be talking about political